Good afternoon all. My name's Sharon Davis and I'm an Enterprise Senior Fellow with the Water, Environment and Agriculture program with the University of Melbourne, and I have the pleasure of being your MC for our webinar today where we're discussing COVID-19 and wastewater surveillance. I'd like to begin though by acknowledging the traditional owners of the land from which I'm participating in this meeting, the Wurundjeri people, and pay my respects to their elders past, present and emerging. Today's event is part of a regular series where we discuss important issues about water security and where we aim to bring together thought leaders from academia, industry and government to provide evidence-based insights and to raise the quality of debate around these important issues. The Water, Environment and Agriculture Group at the University of Melbourne work with industry, government and communities to find innovative solutions to tackle water security and environmental challenges. This is the second year of our highly successful water security series, and our past events have covered diverse range of issues including gender equality, impacts of recent bushfires, diversification of water supplies, environmental outcomes from the Murray-Darling Basin Plan and contaminants of emerging concern. If you'd like to learn more about the program or view any of the earlier seminars, please visit our website and the details of which will be on the screen shortly. Before we begin the panel discussion, COVID-19 wastewater surveillance, I'd like to take a moment to thank you for your participation and for registering in our webinar. We've changed our format to bring this to you online to be able to continue our water security series in 2020. And obviously the new format presents some new challenges, so I would like to acknowledge and thank you for your patience in terms of logging in today. It seems like there might have been some issues. And also share with you that I am having some connectivity issues with my computer at home. So if I happen to lose connectivity, I'm sure everyone has this experience. I'd like to apologise in advance and note that Professor Peter Scales will step in if required. I'd like to also note that this event is being recorded and will be put up on the university's Melbourne School of Engineering's webpage, so it'll be able to be accessed after the event today. And as part of today's discussion, as we do with all of our series, we'll be answering audience questions, and I encourage you to engage as an audience through the question and answer box, which is within the Zoom app and generally found at the bottom of your screen. So you can submit your questions through the Q&A chat box, and then I will moderate those questions and will aim to answer as many as possible with our panellists. If you'd like to participate via social media, you can use the hashtag WaterSecurity20 for Twitter, and we'd be pleased to have you do that. As an Enterprise Senior Fellow, my role includes building a bridge between academia, government and industry, and through those connections and exchange, create more positive outcomes for our sector as a whole. Our water security events are an important mechanism to create and strengthen these connections and to build greater understanding in our sector. And it's my absolute pleasure to be able to introduce the panellists we have today for today's session. First up, Professor Linda Blackall. Linda's the Director, Environmental Microbiology Research Initiative for the University of Melbourne. She's pioneered and championed procedures that reveal microbes and their functions in many natural and engineered systems. And she's been instrumental in discovering microbial contributions to practical aspects of water, wastewater and solid waste treatment, leading to improvement in treatment and energy savings. Nick Crosby is the Recycled Water and IWM Research Manager for Melbourne Water. He's completed a Bachelor of Science Honours at Monash University and a PhD in Marine Biology with the Department of Tropical Plant Science. His research interests include harmful and nuisance algae, emerging water pollutants and waterborne pathogens, and water-related illness. And finally, Dan Deere. Dan is the Water Quality Specialist for Water Futures. He provides independent specialist support to water service providers and environmental health departments on water quality science, management and assessment and certification. Dan provides technical support to various guideline committees covering health-related aspects of drinking, recycled and recreational water in Australia and internationally. So I'd like to warmly welcome our three panellists, experts who we have to talk about this topic today, which we're incredibly pleased to have such a group of people. By way of introduction, the current COVID-19 pandemic is obviously causing great upheaval to society and is anticipated to continue for many months. Our efforts to reduce the spread of the disease are hampered by difficulties in tracking community outbreaks, and obviously complete testing of the entire Australian population is not possible. Today, our panel will explore a different option for virus monitoring: pathogens found in wastewater from toilet flushing and teeth brushing. It may be possible to determine hotspots of viral outbreaks from analysis of wastewater and specific pathogens it contains. And overseas experience would suggest that this analysis is a possible surveillance tool, as proven with other pathogenic virus, bacteria and protozoa. And again, before turning now to the panel, I'd just like to encourage you to take the opportunity to open your Q&A box and please feel free to start submitting questions to the panel. We'll see those come through, so please do. But to get the ball rolling, I'd like to start with a question to Dan, please. Dan, I was wondering if you could give us an overview of the national program that's been put together through Water Research Australia, as well as any other initiatives on using sewage surveillance to track COVID-19 infection rates in the community. Can you indicate where samples are being taken, how often, and who's involved?
Thank you, Sharon. So we've got an initiative that's been set up through a consortium of health, utility and research organisations, and Water Research Australia is helping coordinate that through their members, which include most of the major water utilities in Victoria, the Department of Health and Human Services, University of Melbourne and many other research organisations. And at the moment, we're quite lucky. Victoria has great expertise in understanding how to model and understand its sewage networks, where the sewage comes from, what populations are influenced by that wastewater, where it comes from, how it gets there. It's got people like the University of Melbourne, Federal and Weehai and others that are expert in analysing wastewater. And you've got a lot of well set up water utilities around the state that work well together. And what that means is that Victoria is ahead of the pack nationally in terms of starting up to look at using the COVID-19 monitoring process in wastewater as to inform the control strategies. And so at the moment, there's a couple of processes overlapping. One is to collect samples from wastewater treatment plants and freeze those, ready for analysis later on. And the other process is to work, which is very hard work being done particularly by Vidral and Weehai at the moment, looking at understanding the best methods to use for analysing those samples. And as the case numbers tail off in the community as clinical testing stops revealing anything, and we don't see much, the idea is to use the sewage surveillance as a baseline background monitoring process to pick up outbreaks or pick up hidden cases in the community using a relatively efficient process of testing perhaps a number of sewage treatment plants and other key points around the state, rather than testing everybody all the time. So it's a complementary process.
Great, thanks, Dan. Nick, turning to you now, the surveillance of sewage for pathogenic virus, protozoa and bacteria predominance in a community is an ongoing process by water utilities in Australia. So just not focusing on COVID-19 for the moment, could you describe the current practice in Australia and where it's been useful and what it takes to be able to provide advice to the Department of Health and other regulatory bodies?
Well, thanks, Sharon. Good afternoon to everybody. Yes, I think your question can be answered in three categories. We've been... sorry, I've got a bad habit of being on mute. I think I'm all clear now. Yes, we've been doing this sort of work falling into three categories. One is to provide general information about the number of different pathogens to health departments and also to entities which formulate guidelines which underlie the design of treatment plants. And that's so that we can ensure that our treatment plants are designed for, the most egregious and the most concentrated pathogens in sewage. So we need to have those numbers, and then we build in a degree of conservatism so that all of our treatment plants are very robust, both on the wastewater side and on the drinking water side. So that's one area. The second area is we've been involved with the Victorian Infectious Disease Reference Laboratory, VIDRL, for a number of years now in relation to the polio eradication program that WHO runs. And Australia, being polio-free, has to provide evidence so that we can continue to certify as polio-free. So every year or two, we work with VIDRL to provide samples, and then they can provide that information through to WHO so we can be certified as polio-free. And then the third area is what I describe as more emerging. In recent years, we've been working with UNSW to characterise the number and the pattern and the variety of adenovirus and norovirus in sewage. Now, these are common viruses which underlie gastrointestinal infections. Sometimes they produce cold-like symptoms as well in the case of norovirus. And they're shed at much higher rates than other viruses, and I think we'll get onto that a little later. But they're fairly easy, relatively speaking, to study in sewerage.
Great, thanks, Nick. Linda, now, Nick's been describing surveillance of sewage for pathogens, and I was just wondering if you could please, based on your experience with a range of viruses, tell us about COVID-19, how it's the same or different from other viruses in the context of surveillance.
Thanks for the question. So, as the previous speakers have mentioned, there are already surveillance programs for other viruses, but of course each virus has its own properties that make it more or less easy to actually detect in a water environment. So, just looking at the issues and perhaps also some of the challenges, let me say. So, for example, coronavirus belonging to the COVID-19, rather belonging to the coronaviridae, there are many other coronaviruses that have been sought in water settings or in outside of the clinical setting, let me say. So some of the procedures that are required are present for those viruses. Nick did mention the adenovirus and other viruses that are detected. They are in the gastrointestinal tract, so they're normally present in fecal material and end up at the sewage treatment plant. But the COVID-19 also has been discovered in the intestinal tract as well, and so is potentially going to be shed into the sewage system, into the sewerage system through wastewater, and so can be detected. But the properties, the features that are challenging and distinguish this particular virus perhaps from some other agents that may be searched for in wastewater are the low concentrations, for one thing, and also then the ways of concentrating or trying to look for these viral agents in water, in large volumes of water. So concentration will be one of the biggest features, and then that speaks to the properties of the outside. This is an enveloped virus, so it has an envelope. It sits inside of an envelope, you might say, and so the properties of that envelope are critical for the concentration of it from large volumes of water. So most of the procedures are in place, but there are specific idiosyncrasies that are relevant to this particular virus that will need to be ironed out because of its novelty, its early days in any case.
Great, thanks, Linda. We've had a question. I was wondering if you could answer that, or one of the other panel members. And the question is, how many carriers would there need to be in a particular area for a sewage tester to be able to enable detection?
I guess, well, I'll just take a stab at that one just for now. Look, it's early days, and so the actual the number will be a part of the research that will be ongoing to figure out what will be that number. It can be evaluated using attenuated, or let me say killed, versions of the same virus, so having the same external properties as the COVID-19, so not changing those aspects. So that's where those procedures can be used to evaluate how many viral particles can be concentrated, for one thing, and then detected using the detection methods in the laboratory. But I don't think the answer to that question is known. Nick or Dan may have another comment though.
I could probably add to that, Linda. I agree with you. I think we won't fully understand the sensitivity of this approach for some time to come, and there's a good reason for that, and that is the variability around the world in the extent of clinical surveillance. So we have access to many preprints now in respect to sewage surveillance, and they correlate the numbers they find in sewage with the case numbers that they're reporting. But one has to wonder how good their clinical surveillance has been. So whereas they might claim, say, 1 in 100,000 people sensitivity, one wonders how many infections they had in the community to begin with, which makes it difficult, of course, to come up with robust numbers. And I think we won't know the answer to that question for some time to come.
Can I just jump in with one more comment then? Because the community surveillance is so spotty, that is, we don't really understand the incidence in the community apart from the people who've been tested, which is a very relatively small portion. So that really will take quite a while, and that'll be a hand-in-hand arrangement. That is, as more data for the community carriage becomes available, then so that will speak to numbers in the water as well. Yes.
Great, thank you. I can see some questions are starting to come in from the audience, and I'd just like to encourage everyone, please do feel free to send those questions through. I might just turn to you now though, Dan. You've been tracking other groups around the world, and what have they found thus far that might be helpful to us in Australia in terms of intervention and mitigation strategies, both immediately and potentially into the future?
I think the major message we're getting from the other groups in the world that are working on this, we've got major projects in Switzerland, in France, in Canada, the Netherlands, in various states in the United States. The major message we're getting is we've got to really prioritize very carefully. And there's a range of ways that's important. The first is we've got demand on clinical testing of patients, often using very similar experts, very similar labs, very similar equipment. And we're being told that this mustn't cut across that clinical testing. It mustn't take, I don't know how many of you work in a lab, but there are basic everyday reagents that are used in a lab that are used in clinical and environmental testing. And as Linda said, often the genetic assays that are used are the same, both in clinical and in environmental testing. So we mustn't cut across that. The second thing is, in terms of what we focus our efforts on, we're being told by the health departments there are loads of really exciting research projects that we can do, understanding wastewater, the evolution of the virus, the way it's spread through the community, how it links to things like patient data and so on, just lots of modeling, which is really interesting. But they're saying don't let that distract you from giving us information now to help inform our COVID-19 control strategy. If there's information we can give them now to inform their strategy, they want to see that. That doesn't need to stop the research or get in the way of the research, but they're just saying don't let the research get in the way of the more routine. And the final thing I think is about capacity. There's only so many microbiologists and geneticists and epidemiologists. There's only so many laboratories, only so many samplers that can take sewage samples, only so many water utilities and so on, and reagents and consumables and parts. So as I said, we've got to target our sampling very intelligently and interpret it very intelligently and communicate it that way. As Nick and Linda have just said, these have been used for decades for things like polio, these techniques, and norovirus and adenovirus for many years. But this is a new virus, Hence, we're not really sure yet what the results will mean. So we've got to be very intelligent about how we interpret them. It's not a simple process at this stage. It's going to be quite interesting. So we're all learning as we go. We're all learning together, conducting a kind of real-time live experiment as we go. So
great. We've had a question from the audience actually drilling in a little bit more and asking, what is the actual testing process? Where are the samples taken from, and how much analysis is required?
Yeah, the front end part of that is relatively consistent with other types of testing of viruses in wastewater. So the sewage is collected usually in a litre or 250 ml, 300 ml type volume. So a fairly small bottle that you can carry in your hand, a drink bottle size volume. It's taken usually from a sewage treatment plant where the raw sewage flows, the raw untreated sewage flows in. It can also be taken from other places. You could take it from the septic holding tank of a boat or an aircraft, for instance, as well. So, but it's a raw material, because as Linda was saying, the virus breaks down pretty fast. So there's no point in testing it from treated wastewater. It'll be impossible to detect. And so that gets taken to the lab on ice. It gets held cold so it doesn't break down. It can then get frozen for analysing later or be analysed straight away. And then it goes through a process of cleaning and separating the virus and the genetic information in that virus from all the other rubbish that's in sewage, which would interfere with the test. We call them inhibitors. So you've got to concentrate the virus out, but somehow leave out the inhibitors. And there can be between 1 and 3 stages of concentration and purification, depending on what you're starting with and how contaminated the wastewater is. So that's a real tricky process. That's where it's different from clinical testing. Clinical testing, you've got a consistent sample. With wastewater, you've got a complex sample. You've got to concentrate the material, clean it up. But then the back end of that is relatively similar to what's done in clinical testing. So genetic testing is done to detect, there's only traces of the virus in there. So genetic techniques are used to amplify, to multiply that signal thousands of times so it can be detected. But that's relatively similar to what's done in clinical testing laboratories at the other end of the process.
Great, thanks, Dan. Nick, I might turn to you now. Based on focusing on COVID-19 and building a bit off what Dan's just been talking about, I'm just interested in the COVID-19 in terms of prevalence in wastewater. So what are the risks to public and worker health, as well as laboratory concentration and analytical methods? So what do we know about the virus in terms of its shedding rate from humans relative to other pathogenic virus, and what are the implications for current surveillance programs and ongoing monitoring?
Okay, sure. So taking the first question about risks to wastewater samplers or to consumers of drinking water and the like, as long as people who sample wastewater are wearing the appropriate PPE, we don't have an issue there. We don't see that this is particularly risky, or certainly no more risky than would be the case when you normally sample wastewater. And because the virus is, relatively speaking, quite fragile, it's easily removed through conventional treatment processes. So we have no concerns about the safety of our water or any of the products that may derive from wastewater, for example. Recycled water is an example of that. Now turning to shedding rates, this virus is shed at relatively low numbers. So if you take adenovirus or norovirus particularly is a good example, it might be shed at as high as 10 to the 11 virus particles per gram of stool, whereas COVID is shed between about 10 to the 3, so 1,000 virus particles up to about 10 to the 7 virus particles, maybe a little higher depending on which preprint you look at. So that's a lot lower than the more easily studied norovirus and adenovirus. And the shedding period is also somewhat different. So we've found, looking at the literature, that people are shedding just before symptom onset, but for as long as 6 weeks afterwards. So even after symptoms may have fully resolved, they continue to shed virus in their stool and in other bodily secretions. So Now, I think the third part of the question was, what is the concentration in sewage? Well, that is the question, and we'd love to know. It's a more complicated situation in clinical samples because you have bodily secretions, mostly stool, of course, so when you go to the toilet, but also, you might be blowing your nose or sneezing in the shower, you might wash your hands, they might be contaminated, you might flush facial tissue that you've used down the toilet. So there are a number of ways in which virus can get into the sewage. And because we don't know the number of asymptomatic cases in a population, and that's a very important thing that everybody's keen to understand, we don't really know what levels to expect in sewage, and that will be one of the outcomes of this project.
Thanks, Nick. Look, building off those statements, we do have a couple of questions coming in from the audience about the extent to which a correlation in results of COVID-19 in wastewater can be used to identify particular hotspots. And one of the questions is actually pointing to experience in France where urban wastewater, they're now reporting correlations between spikes in concentrations and hospital admissions in certain regions. I was wondering whether the panel has a view on the opportunities for identifying hotspots through this process or through surveillance.
So Sharon, I might take a stab at that in the first instance, and I think it's a very, very good chance. By the way, we don't know, but we have there are good hopes or good possibilities for using this as an early warning system, and that's exactly how it should be considered. So it's not about, well, we'll find that number of viruses, so there's that number of infections within the community. It's actually, if we found, what in any number of COVID-19 in the wastewater during testing, that would definitely be an early warning, at least for the community. So I think it's a major possibility. The issues need to be overcome, but there are paradigms that we're working off, so it's not like it's completely new. But it can be targeted too to certain regions where you may know that there's a chance of a greater infection within that particular region. Some ethical questions would need to be considered here, but it can be targeted because the sewers are known, their paths are known, and samples can be collected in different locations. So my personal opinion is, if some of the testing, some of the technical challenges can be met or addressed, it will be a major near future and future early warning detection system.
Yes, if I could follow on from Linda, I agree with Linda entirely. We're not at this point in time able to draw a one-to-one relationship between the number of virus particles in the sewage and the number of infections, and there are many reasons for that, one of which is that we don't understand the rate of decay of the virus in sewage. So it will be some time before we can use the data that we collect in a purely quantitative way. But so that leaves us with the qualitative use of the data. And in my view, I think it's shared widely, the most valuable application will be if you are sampling from what is thought to be COVID-free area and you find COVID in the sewage, then that shows that, in fact, you should be looking more closely with your clinical surveillance in that catchment. So that is the most important application. And then second to that would be more of a sort of traffic light approach. So comparing across treatment plants that are fairly similar or sites are fairly similar, are you seeing kind of red rather than green in terms of numbers? So you're not speculating about the number of cases, but you're seeing a much higher level in one area, and that again should elicit further clinical surveillance.
Thanks, Nick. We have a question here that asks whether the presence of the virus in raw wastewater might be impacted by the use of disinfectant and cleaning chemicals.
Yeah, it is, and it is for other viruses too. So even things like norovirus that Nick mentioned are much easier to find. Sometimes there are some wastewater treatment trains where it's really hard to get them to find them, and the treatment plant nearby, it's easier to find them, relatively speaking. So you're dead right. The nature of wastewater affects it, which is why it's a bit of an expert job to take wastewater and detect viruses in that, and why you've got to look at each treatment plant a little bit independently. And for some sites, for example, the term uses trade waste. So if you take a sample from a building or from a cruise ship or something like that, that's a different concentration again. And detergents do break down the virus. Other chemicals can break down the virus, make it much harder to detect or interfere with the reaction. So a lot of inhibitors in environmental samples can make it hard to detect viruses in environmental samples as well. So it's a good question, and it is a big problem. That's why a simple standard method won't work for all wastewater. You've got to be able to modify and adapt to what you find. One good thing, though, is that Vidral's been working on some control viruses that can be added as a positive control. So at least we'll know it's not working. And then we can look at how to modify the method to get it working. So at least you're not going to have the problem of not knowing that whether it's working or not. You'll have that inhibition, that problem will be picked up by the controls.
Thank you. And again, building off that, we have a question. In the long term, once more is known about shedding rates, etc., what are the greatest technical challenges in continuously monitoring for COVID-19? And are you aware of any new technologies that will help with those challenges?
So again, I'm prepared to kick off the answers to these questions. I think the biggest,
Linda.
Yeah, I think that one of the biggest challenges really is getting the virus or its RNA from the wastewater samples, because it's going to be present in such relatively low concentrations, as we've heard, relative to other viruses that are successfully detected. And so the concentration of the viral particles will be will be one of the major features. Once one is confident that you can, find a viral particle because of the sensitivity of some of the molecular, biological, the genetic techniques that are used for the detection, can be tweaked and can be improved in their sensitivity as well. But concentrating the samples in a fashion that obviates contaminants, because in wastewater there's a whole mixture of different compounds, as was mentioned before, the chemicals that we just talked about will be contributing. And so in that really complicated milieu, how do we sift and find these viral particles for the detection? And there are quite a few developments being explored in that region. So that's one of the research areas that's really important and critical. And so I think that's where the major challenge is at the minute. So once obtained in a, let me say, inhibitor-free status, the rest of the procedure will be, there are trains in place for the rest, but there can be modifications there as well.
Yeah, I think, Linda, you're right, and we've had lots of approaches from biotech companies and other providers saying we've got some really new technology that you guys haven't been using in the wastewater sector, that you should try. We haven't really got time to sort of try them all and pilot them all, but certainly Water Research Australia has asked IL Utilities, who's a technology innovation evaluation company for the water industry. They do things like new pipes and pumps and things, but also new technologies, to start looking at those, because they look, a lot of our methods we use are 25 or 30 years old, and it's not that they're wrong, but there are new technologies that may be much better, as Linda says, to do those extractions. And so we haven't got time to try them all, but we'd like to get them prioritized down to a subset that we then have people like Vidral can try them and prove them.
The other thing I could add to that is, certainly on the concentration side and the analytical pipeline side, there are steps that will improve our sensitivity. but we'll also be aiming to optimise our sampling network. We're keen to help in the here and now. That is our main priority, but we're also building a sampling network that will have strategic value in case we have subsequent waves of infection or subsequent seasons of infection. To date, we have been sampling at a widespread number of locations under quite constrained conditions. In that people have been working from home, so we've had to use existing sampling locations. But as we go forward, we're looking to identify where really are the most optimal places to sampling. And one could be, say, in the middle of a network where the traceback effort isn't so great, but you're still capturing a large number of people at that location. So we will be doing some modelling to determine what an optimal sampling program could look like.
Great, thank you. There's a specific question here for you, Linda, I think, which comes back to a comment that you made earlier about the importance of the envelope in relation to the virus. And the question is, why does it make a difference? Is it in relation to changes in RNA fragments?
So the structure of the virus is that it has its RNA. It's a ribosomal ribonucleic acid genomed virus, but that lives inside of an envelope. And of course, the envelope is where a lot of the immune responses occur. This is where much of the intrigue and discovery in relation to why is this virus different to other coronaviridae, for example. It doesn't make it different to a lot of other viruses, but it is enveloped, and so we do need to be aware that that envelope has chemical properties that need to be considered in the concentration steps, like does the viral particle attach to certain substrates within the concentration steps? And so the physical nature of that envelope is important when looking at concentration. So you don't want to lose the viral particles at some part of the concentration step because it adheres strongly to some aspect of the concentration process and doesn't come through in the liquid that would then be used for doing the testing. So that's why the envelope is important, that it be considered in that concentration step. And I think in relation to the concentration, just harking back to the responses before, it's also to try to increase the volume of the water that's actually able to be passed through to be concentrated. Dan was talking about 300 millilitres. In relation to the amount of water that comes through the sewer, that's a minuscule amount. So if that can be increased in its volume, then that will perhaps speak to the concentration issue. So that's what the envelope's about, is about the physical properties that it may have.
Great, thanks, Linda. We have also a question from our participants in relation to the virus transmission through toilets and toilet reticulation system. Can it be transmitted through sewer vents? I think this happened in Hong Kong during SARS. Would someone like to address that?
Yeah, there's no logical reason that it couldn't theoretically be. That was a highly unusual scenario, and it was linked to a lot of aerosolization of sewage in a very large high-rise building, and then the sewer vents lost their water. It sits in the U-trap, the U-bend, so they became connected to the sewer pipe and got sucked up by vents and caused dozens of cases in the immediate building, but also nearby buildings where the air flowed. And there's been a lot of work done on that, both in Hong Kong and by some stimulation experiments in the US, saying that looked like that is what happened, that there's quite a plausible transmission pathway. But it's I suppose you call it a freak event. It's not something you'd expect to see. There's been nothing else like that. So at the moment, from a practical point of view, we aren't concerned about transmission from sewage to humans. We're concerned about human-to-human transmission, but it is theoretically possible to do that. And the SARS-1 2003 Amoy Gardens outbreak does appear to have been linked to that, but it's direct transmission almost, because it's immediate transmission of fresh aerosols from a toilet straight into the air. There's no time for it to mix with sewage. There's no time for it to start breaking down in the detergents that are in sewage and break up the envelope that Linda talked about. So it wouldn't be a typical scenario. And don't forget as well, normally in that scenario, you'd smell the sewage. And so, it's not, wouldn't happen in low density environments. It would only happen in a peculiar environment like that. But it is a possibility.
Thank you. Great, thanks. Now, we've got a few minutes left, and I just wanted to encourage the audience. I think we've made it through most of the questions we've received so far. If you'd like to submit a question to the panel, please do so now, and we've got a few minutes to answer more questions as they come in. Nick, I guess in the interim, I wanted to turn to you and test from what we've heard. There are some challenges with tracking the prevalence of COVID-19 when you compare that with other viruses. Do you think it's still worth doing if we can't see, if we need to have reasonable numbers, if we need a pretty high proportion of people to be able to pick up the virus? What's your view on that?
Well, I think the two parts to that. First, if there are a reasonable number of cases in a catchment, you still won't know how many asymptomatic people you have, and so it's valuable to get that insight from sewerage and to compare between catchments where they can be compared. So you might find, you have, say, God forbid, 1,000 people infected in a catchment, but in actual fact there are many fold who aren't showing up to screening facilities. So it gives you some insight there. So yes, it's worthwhile doing that even when you have a large number of cases. And then I guess the second answer to the question is that we're trying furiously behind the scenes all around the world to optimise our process so that we can detect at the lowest number of particles in the sewage, because that is where the greatest gain will be made. If you're able to determine that you have circulating virus in an area where you thought you didn't have any, then that, of course, is very valuable information. And if we get to that point, and I'm cautiously optimistic that we will, then it's a very efficient way of screening, because you're essentially sampling from a large population with just, one sample point or several sample points. So technically it is a high-risk project, but on the other side of the coin, the gains could be very substantial.
Great, thank you. There's a question here from our audience. Is there any area or zone in wastewater treatment process where the virus could hold up for a longer period of time without being treated?
We don't think so. The work on coronaviruses that's been done, as Linda says, there are a lot of coronaviruses. I'll give you some example. When fresh coronaviruses are grown up and added to sewage, we lose 3 log within the first few seconds just by the phase transition from being in one container into sewage. I mean, they're 99.9% killed just by that phase transition. So that the idea of an infectious virus being detected, and that they're very hard to grow and detect and grow in sewage. So when we do detect live infectious viruses, there are only a handful of viruses we can actually detect in a live form from sewage at all. And coronaviruses are very hard to detect in their live form from sewage. They don't live very long. And even the RNA, Linda talked about detecting the RNA signal. People generally aren't finding the RNA or the genetic signal in even the most basic treated sewage, even after it's been treated for a few days, or stored for a few days. So if you put it in the fridge, you might find it for a few weeks. But at room temperature, these viruses will last for a matter of hours to days at the most. even in clean water. And in sewage, where you've got usually a lot of detergents, you think about washing powders, soaps, shampoos, shower gels, industrial detergents, they don't physically stay infectious for very long at all. So we don't expect to find infectious coronaviruses in anything past the most raw sewage. And even then, it'd be very hard to detect them as infectious. We're only really looking for the genetic remains of the virus, not the live. In fact, we considered it early on, going back to sort of the first discussions in February, January. We thought it would basically be a lost cause trying to find infectious coronavirus in wastewater, because previous work with other coronaviruses had found it's just too hard to detect them as infectious form. They break down too fast.
Great. Thank you.
Can I just follow up on that a little bit about the infectious nature of the virus that you may well detect in wastewater? Actually, detecting non-viable or non-living virus is equally useful, because it's, like any detection of any sort in wastewater tells us that this virus is somewhere in the community, and that's what Nick was talking about before, the traffic light, the green or the red or whatever. If we did see something, it definitely will not be infectious, as Dan has just described, but it still tells us about something that's coming from the human population, from the community, and so it's super valuable.
Great. Thanks, Linda. Look, one last question from the audience before we wrap up. When is a system of sampling likely to be up and running in Victoria?
Yeah, I can take that one. Yep, so we've been sampling in Victoria since the 1st of April, and we're sampling at quite a number of sites within the metro area, about a dozen in all, and a similar number of wastewater treatment plants, mostly within 200 kilometres of Melbourne, but some other more abroad sites, including at locations where where the clinical surveillance tells us there aren't any COVID cases, and they're our control catchments. And we hope to commence processing those samples very soon. We're in the final stretch of optimising our methodology before turning to screening of samples. And we don't, as Dan alluded to earlier, we don't want to wait too long, because we would need to get onto the testing ASAP so we can provide preliminary information back to the health department.
Great. Thanks, Nick. Look, in the interest of time, I think we'll have to leave our panel discussion there. So I just want to take the opportunity now to really thank our panel members and participants in our session today. It has really been an incredibly interesting discussion on this really important issue, and I'd really also like to thank our virtual audience. We can see that you're out there, but can't see your faces, obviously, in terms of providing such thoughtful and thought-provoking questions in to the panellists, which has been really great to help drive the discussion. There's a couple of things that I just wanted to share with everyone before we wrap up, and that is that our next water security event will take place on Wednesday, the 17th of June. And we'll be discussing the changing nature of the water energy nexus in urban water supply systems. So to register, please go to our website and keep an eye out for the event in coming days. But we'd also like to thank you again for your participation, and we'd really appreciate your input to a post-event survey which will appear on your screen when this session ends. We'd really value your feedback to help us shape the future of our series. If there are topics that you're particularly interested in, or people you're particularly interested in hearing from, we would really like to know. It's an objective of ours to ensure that we're that we're helping support more informed debate around water issues. So we'd really appreciate your feedback. So with that, I've actually scooted through that quite quickly. So we do have a minute or two left. Do any of the panellists want to make any closing comments before we end the session?
I think the only comment I'd make, Sharon, is that these kind of technologies are, I suppose, they're expert systems. They need expert interpretation, expert delivery, and expert usage. And in those correct hands, they are very useful, potentially very useful technologies. But equally, if they're misinterpreted or misused or done badly, they can give confusing, misleading results. So that's why I think you'll find the sort of stuff University of Melbourne's been doing and the groups that people like Nick and others have been working with, they've tended to work with those that have the most expertise in this area and the most experience to avoid potentially making some mistakes and having some misleading results. The other thing is when people criticize the flaws in these methods, and there are plenty of them, don't forget there are also flaws in clinical methods, there are flaws in contact tracing. All the tools available in the toolbox to manage this pandemic have flaws. So this is one more tool. It has its flaws and its weaknesses, but also has its strengths. So if intelligently used with the other tools, it should provide useful information.
So I just have one final comment as well. I mean, like with the development of the vaccine for the COVID-19, many laboratories in the world are using untried methods in an effort to quickly come up with solutions. And that's the same here as well. So thinking outside the box to try to, as Dan has already said, there are companies who have said particular technologies are not used in the wastewater industry. And others are thinking outside the box as well. So I think this is a really good forum because quite a lot of people probably listening and they can contact panellists and go forward because I think it's going to be a really interesting future in the area. There's lots of options, but I absolutely concur with Dan. Nothing will be absolute, but the traffic light system that Nick mentioned earlier, I like that very much. And it is there anything or is there not anything detected?
Thanks, Linda. Okay. All right then. Well, just on 1 o'clock now, I guess I will call our seminar to a close and thank all of our participants again for your involvement and your interest, and we look forward to seeing you again at our next water security series. Thank you, and thanks to the panellists again. Great, wonderful.